Hello guys, welcome to another video of BioWorld. So in this video we'll be seeing about E. coli. E. coli is a type of bacteria which causes UTI, that is urinary tract infection. Okay, so we'll be seeing about the morphology and its diagram, pathogenicity and its cultural characteristic, diagnosis and treatment of E. coli in this video. So let's get into the video. First we'll be seeing about the morphology. Okay, so E. coli is a type of gram-negative rots. Okay, so it occurs singly or in pairs. When we culture it, it will either occur in single form or in pairs. Okay, so the size of E. coli will be 1.3 into 0.4 to 0.7 μm, and it is motile by Petritrichus flagella. So once you see here is this is the Petritrichus flagella, which helps in the motility of the organism. Okay, so it is non-spore forming and normal flora of intestinal tract. It is a commensal pathogen of intestine in humans. Okay, so it protects intestinal tracts from other pathogens. So it will protect our intestine from other pathogens, which is invading it. Okay, so you may have a doubt that then how E. coli will be a pathogen. So E. coli will be a pathogen once it rises in population. Okay, so unless and until it rises in population, it doesn't cause us any disease or infection in intestinal tract. Okay, so that's all about the morphology, and we'll be seeing about the diagram. So diagram, this is the structure of E. coli, that is rod shaped bacteria. It has an outer membrane and cell wall, periplasmic space. So here in periplasmic space, that peptidoglycan layer will be present. Okay, so inside it will be a DNA DNA of an organism present, and around that ribosomes will be present. So what is the role of ribosome in a cell means it helps in the protein synthesis. So obviously ribosomes is very essential for a bacterial cell. Okay, so the small hair-like structure in the outer membrane is called pili. Pili helps the bacterial cell to adhere to the intestine. Okay, so that is the role of pili in the bacterial cell. Okay, so and the black colored dots have projected here is a toxins produced by E. coli. Okay, so this large one is the Petritrichus flagella, which helps in the motility. Okay, so this is all about the diagram of E. coli. Now let's see about the cultural characteristics of E. coli. So it is an aerobic and facultative anaerobic. So it grows well in presence of oxygen as well as in the absence of oxygen. Okay, so it is both aerobic and facultative anaerobic. The growth temperature of E. coli will be 37 degrees Celsius in 24 hours. Okay, so it needs 37 degrees Celsius for its growth, and in fresh isolates smooth forms will be seen, and in older isolates rough forms will be seen. Okay, so then some strains of this E. coli will be seen in mucoid form. So some only certain strain of the E. coli will be seen in mucoid form. It grows well in nutrient media. Okay, nutrient media is rich in nutrients, so it grows well in nutrient media. Okay, so now let's see about the pathogenicity of E. coli. So what type of infection it causes means urethritis or cystis. Urethritis is an infection which is caused in the urethral region. Okay, so urethritis is a type of infection which is caused by E. coli, and mostly 90% of UTI is caused by E. coli. Okay, so comparatively other bacteria, this E. coli is main reason for causing UTI. Understood? So next we'll be seeing about its pathogenesis. Okay, so E. coli as I said already, this pili, small hair-like structure, will get adhered to the enterocyte membrane of the host. Okay, enterocyte membrane is nothing but the outer membrane of the intestine. Human intestine is known as enterocyte membrane. Okay, so E. coli uses its pili to attach to the enterocyte membrane of the host. Okay, then after attaching to the host, it translocates. Okay, translocation will happen. So bacterial TIR translocate to cell membrane of the host. Okay, so the bacterial TAR will translocate into cell membrane of the host for E. coli to attach firmly. So for its firm attachment, bacterial TAR will translocate to cell membrane of the host. Okay, so then after it's firmly attaching, then its process will be binding. Okay, now this E. coli binds with TIR to firmly attach. Okay, now after attaching, this toxin will be released from the bacterial cell, that is Shiga toxin will be released. Okay, so after that, some receptors like GV5 and GV receptors are stimulated and toxins gets bind with it, and after that toxin are internalized into the cell. Okay, released toxins will get internalized into the cell, and obviously it stops the protein synthesis. It will completely destroy the ribosomes, as the ribosomes is responsible for protein synthesis. So this after that, if the protein synthesis of a cell is completely stopped, it results in the cell death. So that is happening there. Okay, so cell death is happening there as ribosomal synthesis is completely stopped because of the toxin release. Okay, so now as the cell becomes dead, it results or it causes bloody diarrhea in the host. Okay, so that is the reason for the bloody diarrhea appearing in the host. Okay, now this released toxins will get into the circulatory system. So as the blood circulates all over the body, it will get transmitted by the blood inside the circulatory system, and obviously it will damages the RBC, platelets and other organs like liver, heart, and all other organs will be damaged. Okay, not only the organs, these RBC and platelets will also get damaged by the toxin release. Okay, so that's all about the pathogenicity. Now let's see about the lab diagnosis done for E. coli infection. Okay, so first and foremost lab diagnostic method is gram staining. We have to do gram staining for identify which type of bacteria is present, either gram positive or gram negative bacteria is present. So for identification of that, we have to do gram staining. So in gram staining, the results will be gram negative rods, and specimen will be taken here is urine sample. Okay, so urine sample will be taking, and after all the procedures done, we'll get a result of gram negative rods. Okay, so which type of media we'll be using here is McConkey agar, blood agar, nutrient agar. Okay, so as E. coli is a glucose fermenter with acid production, we'll be using a McConkey agar. So if we use McConkey agar, we'll be seeing mucoid pink colony. Okay, so as E. coli is lactose fermenting, we'll be able to see mucoid pink colonies. If we culture this in blood agar, we'll be seeing moist, non-creamy, white colony. Okay, if we culture in nutrient agar, we'll be seeing colorless colonies with smooth, large colony. So this is all about the lab diagnosis done for the infection of urethritis. And now let's see about the treatment done for this type of infection. So the treatment here is cephalosporin, ampicillin and doxycycline. Okay, so this three is the foremost or first line treatment, antibiotic treatment given for the urethritis infection. Okay, then we'll be using trimethoprim and sulfmethanonazole and B lactame. So these are all the first line antibiotics we'll be giving to the patients who is infected by the urethritis. Okay, so next let's see which type of symptoms occur in this disease. Means mainly pain occurring areas will be bladder, groin, lower abdomen. Okay, so in this areas pain will be seen. And during which circumstances this pain will increase means during urination and sexual contact. So both during urination and sex will feel more pain in this areas. Okay, which areas means bladder, groin and lower abdomen. And urine will be like, it will be foul smelling urine and dark urine will be seen. Okay, so persistent urge to urine. So patients who are caused by this type of infection will urge to pass urine frequently. Okay, so that is a main symptom seen here. And the whole body, if we take the whole body symptoms means we'll be having fatigue, fever, cramping in abdomen and vaginal irritation. In female we'll be seeing vaginal irritation. Also in some cases the vaginal discharge also will be slightly varying from the normal persons. Okay, uninfected persons. So this is all about the symptoms. And how we can prevent means staying hydrated is the best option. Okay, so stay hydrated, urinate after sexual activity. So after having sex we have to urinate so that we'll come out of the infection causing by the UTI. Okay, so next we have to empty bladder often. So often we should empty bladder with persistently and maintain hygiene. This maintaining hygiene will help a lot. Okay, so then wipe front and back after urinating. So this all comes under the hygiene. So we have to maintain proper hygiene to escape from this type of infection causing. I hope you enjoyed the video and the video was useful. Thank you.